Okay, good morning and welcome everybody to Gem Pharma Tech's monthly webinar series. My name is John Jackson. I'm part of the marketing team here. And today we're going to hear from one of the leaders of our metabolic pipeline, Dong Iran. She's going to share a little bit about the expanding indications for GLP-1 receptor targeted therapies in mouse models of metabolic disease. Before she does that, I'm going to explain a little bit about our company and what we do. Very briefly, our mission is to engineer a healthier world by pushing the boundaries of biomedical innovation. One of the ways we do that, obviously, is by engineering thousands of mouse models. And on the other side of that, we provide the services that basically deliver, the data and insights that moves research or drug discovery forward. To share a little bit about how we're set up organizationally, we have 8 sites worldwide, including a new headquarters in San Diego. As you can see here, where I see a lot of people who have signed up, have EDU addresses, which we can move our models through San Diego as well. Speaking of models, we have over 25,000 genetically engineered mouse models. These are independently developed, and we have the capacity to generate 6,000 new strains each year. So we've really positioned ourselves as experts in gene editing and providing models to scientists who have these target genes of interest to do their research. On the kind of CRO side of things, which we're not going to focus on today, if you'd like to learn more, we're going to put up an email address at the end of the presentation. But we've completed over 1,000. We complete over 1,000 preclinical studies each year, across all of our sites, and we've done studies in every disease area you can imagine, whether it's neuroscience, autoimmune, oncology, rare disease, or like today for metabolic disease. So that's who we are in a nutshell. And today, obviously, Iran is going to share a little bit about what we've been doing in the GLP-1 area. And so I'll turn it over to her. She's going to go through a little bit of background information and case studies. And at the end, we will have a Q&A session. But I'll let her explain a little bit more about what she'll be talking about.
Yep. So just morning, everyone. So for today's talking, I'm gonna divide this into 3 main parts. So for the first part, we're gonna give a background information on the current stages of the GLP-1 landscape, including some market sales and also some future forecast. And then for the next part, I'll give you some detailed cases, including some indications of the GLP-1, including some initial indication of the GLP-1, the anti-obesity, and then also with some expanding indication, some chronic kidney diseases, and MASH model, and also the half-path mass model. And then it will be our last session, the Q&A session. If you have any interest or questions, then we could have a discussion on that part. So for the first one, I'm gonna give a snapshot of the GLP-1 drug market performance. So actually for the GLP-1 kinds of drug, it's a really popular kinds of drug for these several years. So when we focus on this year, the 2025, for the first 3 quarters, we could see that the Lilly's tirzepatide, the double target, the GIP and the GLP-1 double target kinds of drug, and the Novo Nordisk single target, the semaglutide GLP-1. They do also show the incredible drug market performance. And from the first 3 quarters, they average for around 24.8 and for $26 billion for each drug. And then we give a whole year forecast from the 2025. We could see that for these 2 drugs, they could reach around $33 billion. So here we could see that for the GLP-1 kinds of drug, they really just reach success drug market performance. And when we just try to doing a prolonged forecast, when we go into the prediction on the 2033, you could see that the forecast of the overall GLP-1 kinds of drug could reach around $56 billion. But we could see that the drug, GLP-1 drug, was not only for the single target or also for the double target, the semaglutide, and also for the uh, tirzepatide. They also are focusing on some multiple target combination therapy, including some retatrutide, the triple target, and also maybe some combination used by the amylin. So the future trends maybe is the combination of different targets with the GLP-1, and also the expansion of the expansion of the indications based on the GLP-1 treatment is also the future trends. So why it's quite popular of the expanded indication, we could just back onto the mechanism of the GLP-1 kinds of drug. So for the initial indication of the GLP-1 use was some anti-obesity, anti-diabetics, but we could see that for the organ targets, the GLP-1 kinds of drug not only target on the pancreas or some fat tissue. They also have some targets on some kidney, intestine, heart, or some other organs. So this is why we are trying to using the GLP-1 to doing some expansion of the indication. So here we could using the semaglutide as one example of the GLP-1 kinds of drug. So here we could see that for the expansion of the indication, the semaglutide can not only using in the weight loss, this field, they can also just doing some expansion on some cardiovascular health and some liver health, and also maybe for some cancer because some different expanding indication. So here is the evidence of how we doing some expansion of the GLP-1 indication. So here we could see that first is for the obesity and for the type 2 diabetes, the GLP-1 treatment have already get the approval of the FDA. And then for some expansion of the indication, the MASH, diabetic kidney disease, and also some cardiovascular disease, and the sleep apnea, they all got the approval from the FDA. And then other indications like the PCOS, they are all in some clinical trial, but in different phases. So here I'm gonna to just talk about some details about some cases of the expansion of the indication. So I will mainly focus on the obesity, diabetic kidney disease, MASH, and the HFpEF. So the first part I'm gonna go through about our initial indication of the GLP-1. So for the initial therapy by using the GLP-1 is some anti-obesity kinds of drug. But when we're trying to using the semaglutide, we using this as an example, we found that the semaglutide, yes, it do show some weight loss effect, but it do also show some lean mass loss. So here, how we could just to have a relief or some improvement of gain muscle is an important issue for us to be solved. So here we could combine use with some other drugs like some Bimagrumab, and then how to just trying to improve some energy metabolism is another issues we need to solve. Then we could combine with the MGL 3196, the THR-beta agonist. And then also, we could also combine it with other target kinds of some amylin, and not only for this kind of drug, we could also combine it with using some SNRI kind of drug. So, and also, expect for the combination used, we could just explore some more different pharmaceutical of the semaglutide, like some oral one, the Rybelsus, alpha-lipro, and also some triple target and some other targeting. So these all are some trends or some future strategy to reach the future weight loss by combined with the GLP-1 kinds of drug. And here, not only for some clinical trials. So the preclinical mouse model is very important for the preclinical drug development. So our Gene have also developed some mouse model in order to in line with some clinical request. So here we have developed a base mass model. We have using the mass induced by the high-fat diet. And then based on this mass model, we have also tried do some drug treatment and to see some drug effect. So for this first case, we have using the Wegovy, which is the inject semaglutide, combined with our muscle gain drug, the Bimagrumab. Then from the drug efficiency results, we could see that this combination use drug could show a larger window for weight loss compared with our semaglutide single used. And then this combination could also show some delayed muscle loss. So this combination could not only gain some weight loss, it could also show some enlargement of the GLP-1 drug efficiency, a bigger large window of the weight loss, and then also solve the problems of the lean mass loss. And not only for this, we have also combined the semaglutide treatment with the THR-beta agonism. So here for this, not only for some body weight monitoring and some body composition monitoring, we have added a metabolic cage detection. And then we could see from the results that this combination used could have a larger window for weight loss, and also it could delay the muscle loss. And then from the metabolic cage results, we found that this combination used could have improvement on some energy metabolism. And not only for the anti-obesity one, we could also just to doing some expansion of the indication. So the first expansion indication was the GLP-1 receptor agonism on the chronic kidney disease for this field. So the first part is how is the clinical patients shows type 2 diabetes. So for healthy people, when some people could have some food intake, maybe they have a delicious meal, their glucose may increase. Their glucose will increase. And then our pancreas will secrete the insulin, trying to decrease their glucose levels. But for the type 2 diabetes patients, they could show some resistance on the insulin, which cause the insulin resistance. And their organs and muscle don't have a quite sensitive response to the insulin. So this is caused the type 2 diabetes. So how does the GLP-1 could have a treatment on the type 2 diabetes? Is for the as we just talked, the GLP-1 has a multiple organ mechanism. They could being act on different organs, but the mainly efficiency on how to treat the type 2 diabetes is on how the GLP-1 could increase the insulin sensitivity. They could have some increase the insulin secretion and also could decrease the glucagon secretion. And then by other mechanism, the GLP-1 could have a relief on the insulin resistance on the type 2 diabetes patients. And here is some newly approved of the GLP-1 using on the type 2 diabetes patients. Actually, the FDA have approved one kind of the injection semaglutide, the GLP-1 receptor agonism. Then it's also proven that the GLP-1 could reduce the rates of worsened kidney disease in the adults, which they have already got the type 2 diabetes and some chronic kidney disease symptoms. And then just before this meeting on the October of the 2025, the oral semaglutide was also approved for some type 2 diabetes patients. And this is just some another expanding indication is a cardiovascular disease. So for both different administration route of the semaglutide, they all got the approval from the FDA based on the type 2 diabetes. And also, as we cite, not only for the clinical, we also have developed this. There is also some classic mass model for some preclinical drug development. And so here we just give an example by using the BKS DB mass model. So for this is BKS mass model, it was we have this mass model shows some model leptin receptor deficiency. So it's a spontaneous type 2 diabetes mass model. So here we could see some phenotype of this mass model, and we could see that the mice shows some higher body weight and also shows higher blood glucose levels. And the hemoglobin also is higher compared with our health mouse. Then, based on this classic mass model, our Bimagrumab also have to do some drug efficiency to to see whether it could be in line with some clinical data. And then this whole drug efficiency period is 4 weeks, and we have evaluated some phenotype, including their body weight, food intake, blood glucose levels, and also some other blood parameters. So here is this slide shows some body weight and food intake. So in this study, we have using some GLP-1 related therapy. So we have using the tirzepatide, retatrutide, which is one is the double target, and then the retatrutide is the single target. And then we have also add one positive drug, the empagliflozin, as a comparison. So here from the body weight data, we could see that this triple targets the GLP-1 therapy, the retatrutide, could have a significant decrease on the body weight. And then this kind of drug is also shows some effect on the food intake. And here was for it features several important parameters on the type 2 diabetes mass model is the blood glucose levels. You can see that both for the postprandial and also for the fasting blood glucose, the GLP-1 related therapy, the TZP and the retatrutide could all have a decrease of their blood glucose levels. And the same trends could also see in the hemoglobin levels for both our GLP-1 related drugs and also for our positive drugs. They also shows a significant decrease compared with our non-treatment, the vehicle group. So not only for the blood glucose levels, when it comes to the urine biochemistry levels, we have detect its UACR levels. And we could see that for the double target TZP, triple target retatrutide, they all shows a significant decrease compared with the vehicle group. And not only for the type 2 diabetes mass model, our Bimagrumab also have developed some other chronic kidney disease mass model, also found some diabetic nephropathy. So based on the BKSDB mass model, we have do some gene editing to do the deleting of the NOS3 gene. And then we have also plus some virus about the MAPS renin virus to induce mice diabetic nephropathy mass model. And then we also have doing some surgery by cut one side of the mice kidney to try to build a chronic kidney disease mass model. And then not only for the diabetes and obesity, the GLP-1 receptor agonism kinds of drug have also doing some improvement on the liver health, which we could go to the MASH syndromes. So here is the progression of the MASH or some liver disease. So from the beginning, people will just show some fatty liver, and then around 12 to 40% patients will be going to the MASH. So the MASH, they will show some steatosis, inflammation, and some bloating, and some of them also show some fibrosis. And then after the MASH, the next stages will be the cytosis. And after cytosis, there will be some HCC, some liver cancer. So why it's quite important, why it's critical to using the MASH or to doing treatment on the MASH is because after this stage of MASH, so the progression of the disease is irreversible. So means that it's critical to doing some drug treatment on this MASH model. But there is several risk factors on these MASH patients, like some obesity, type 2 diabetes, insulin resistance, and maybe some other liver lipid levels increasing. So there was quite a lot of risk factors that could cause into the MASH. So also the drug treatment is also important, not only for the lifestyles changing, also we need to doing some drug treatment. Here was some clinical trials of the MASH. So here we could see that up to now we have quite a lot of different kinds of drug, kinds of some THR-beta agonism, GLP-1 receptor agonism, also some FGF kinds of drug, this family. They all was in some clinical trials that we are trying to find whether they could have a relief on the MASH syndrome. And the results was yes, for both the fibrosis and for some MASH resolution, they all show some drug efficiency. So maybe for the GLP-1 receptor agonism for this topic, how does this kind of drug could have a relief on the MASH? So actually, this kind of drug was mainly could decrease the accumulation of the fat liver lipids and also same as some treatment on the diabetes, it could help some insulin secretion. And then also for some anti-obesity effect, it could help people to decrease their appetite. And so for this different mechanism, this kind of drug could have some body weight loss, blood glucose decrease, and also shows a relief on their fatty liver or MASH effect. So not only for the GLP-1 kinds of drug, the THR-beta and some PPAR agonists, they also could have a relief on the MASH models. So their pathway was through some fatty acid oxidation. So we could see that the MASH treatment could be approved maybe by different pathway. And then maybe we could move on to some clinical status of the GLP-1 on MASH model. So here we could see that the MASH, the FDA have already approved the semaglutide, our GLP-1 receptor agonism kind of drug to treatment on the MASH. And then, there was approved that the patient with the moderate or advanced with the MASH could have some resolution of the steatohepatitis, and there was no worsening of the liver fibrosis. And based on this, we also have some preclinical mouse model. And here we using some we have first using a high-fat induced mouse model. It's a obese mouse model. Then we have plus a chemical toxin, the CCL4, to build this mesh model. And based on this, we also have doing some drug efficient tests. So for this drug efficiency, we have using the FDA approved drug treatment, which is the MGL, the 3196, the THR-beta agnosis, and also our GLP-1 related therapy, the tirzepatide. So in this clinical trial study, we have just evaluate their body weight, and here we could see that our tirzepatide could have a decrease on the body weight, and also they could have some decrease on their food intake amount. When it comes to their liver function, you could see that both for the tirzepatide and also for our THR-beta agnosis, they could all significant decrease the ALT and AST level. And when we're doing some liver lipase evaluation, they also some significant decrease compared with our vehicle group. And for the endpoint, we also collect the liver from the mice. And when we're doing the evaluation by using the HE staining, we have doing a scoring by using the NAS scoring. And here we could see that our tirzepatide and also our THR-beta agnosis, they all shows significant relief on the mice pathology, including steatosis, also for some inflammation and blooming. And this is the fibrosis. We have doing some PCR staining and to do some fibrosis evaluation. And here we could see that our THR-beta agnosis could have a really significant decrease on the mice liver fibrosis level. But for our tirzepatide, we didn't see a significant decrease. So this is also in line with the clinical trial results with the no worsen of the liver fibrosis stage. And not only for this high-fat diet plus the chemical toxin one, we have also have some diet-induced one by using the gun diet. Also, we call this the Western diet induction. And except for this, we have also some gene editing mouse model, which we have called this B6 ALMAS1 delete mouse model. So here we could see that they are showing different disease stages for both different liver fibrosis, and also this different mouse model could also by using different kinds of drug. So means that for a preclinical drug development stages, a mouse model chosen is very important. And then we could move on to some heart relief. So the GLP-1 receptor agnosis also could be used on some half-path. So here is some clinical data. We could see that after the treatment of the semaglutide, compared with the placebo group, we could see that the dies inducing by some cardiovascular diseases or some worsen of the heart failure event is decreased. So for this red line is the placebo group, and then for this one is our GLP-1, the semaglutide treatment group. So how does the GLP-1 agnosis could have a drug efficiency on the half path? So here we could first the GLP-1 could decrease the atrial enlargement, and also it could have decreased the epicardial fat. And also, it could increasing some metabolism ability on the myocardial glucose, and also it could decrease some cardiomyocyte apoptosis. So based on this one, we also have some preclinical mouse model. So here we have using induction by both using the agonist and also the high-fidelity induction. So the total period is 10 weeks. And here we have doing some evaluation and to see whether this mouse model is in line with some clinical standards. So from the mitral inflow and some Doppler tissue curves, we could see that our name and high-fidelity induced half-path mass model is in a moderate and a severe dysfunction stage. So here we also by using some echocardiography and to see some parameter, and then we could see that they all reach the standard of some clinical standards, and then the ejection factor is remained. So this is also in line with the half-path mass model. And also for the blood pressure, we have tested, yes, this mass model also shows a higher glucose level compared with our CHODES group. And then we also have doing some evaluation of the heart. We could see that there were some in-situ fibrosis compared with the normal group. So this heart fibrosis is also in line with the clinical standard. And then based on this mass model, we have also conduct a drug efficiency, and here we have using the DAPA and also the TZP to see their drug efficiency. And here we could see that they don't have a quite effect on their ejection fraction. Yes, because for this mouse model, this is remaining. And then we found that these kinds of drug, our DAPA and TZP, they all could have some improvement on the diastolic and systolic functions. And so based on this, we also have some other mass model, including some induction one and also some surgery. And we could see that we have different time period, and then you could, it will be helpful for some preclinical drug efficiency development. So that will be all for today's talking. We have just give some briefly introduction on the on some expansion of the GLP-1 indication. And also we could see that based on their multiple organs mechanism. So we the GLP-1 kinds of drug could have maybe a quite potential of both for the maybe markets or some other expansion of the indication. So just let's see. So I think that's all for my talking. If you have any questions, you could tap on the chat box or maybe on the Q&A section, then we could have a talk on that. Yeah.
As those questions start to come in, first I'd just like to address, yes, this webinar will be available on demand. All of our webinars are available on demand. If you go to our website and go to the resource section, there is a webinar subsection under that where you can access this webinar and any of the webinars we've done in the past. We'll give a few more moments for more questions to come in.
Yeah.
If you don't feel like typing your question in or having it answered live, or maybe you have a question unrelated to any of the topics presented today, you can always reach out to sales@gempharmatech.com, and we can have somebody answer your questions right away. We have one interesting question that's come in, Zhang Ran, how many weeks are the
Mhm.
B6 mice on the diet before they become obese?
Mm, Actually, from our historical data, we should around 10 to 12 weeks old. So we will define the obese mass model at around the mice body weight at an average value at over 45 grams. So we could give the high-fat diet induction for around 10 to 12 weeks. Then it could reach this standard.
Is that standard across, everybody who makes these mouse models, or is that something special for us?
Oh, sorry,
Is that
just
the standard method? I was asking if that's the standard method, no matter where you get your mouse.
actually for the standard because we need to give some drug efficiency window, so we need to set the standard to make sure that it's reach a base level.
Okay,
Yeah.
another question.
Mm.
For the diabetic kidney disease models, what's the
Mhm.
difference between the AAV rennet and the NOS3 deletion models?
Mm, Actually, there are different mouse established method. So for the nursery, it's a gene editing one. So we based on the BCSDB mouse model, and then we have deleted the nursery mouse model. So this one is for some genetic level. Then for the mRNA one, we have plus of virus to do induction on that one. So this will be some two different ways. So actually, maybe it's more important on people how to choose these different kinds of drug, these different kinds of mass model. So it's based on maybe it could based on the uh clean as clean as mice mechanism. If your drug pathway will go through the NOS3 one, because for the NOS3 one, we have already deleted that string. So we recommended to using, maybe we could recommend it to using the app-raining one. So that will be more suitable for. So how to choose the mass model or the differences maybe based on the claims or people's drug efficiency or drug mechanism.
A
Uh,
few
I
more.
think I got another online question was because for the diabetes and the obesity, maybe we could back to the slides because yeah, yeah, our diabetes mass model was all based on the obese mass model, the BKSDB mass model. So let's go on this part. Yeah. So here we could see that except for this one, this mass model, all the BCAS-DB or NOS3 delete one and also the mRNA induction one, they are all show some obesity.
Okay, a
Yeah,
few
so
questions in the Q&A section that are very
mhm.
specific to treatment standards.
What's the
Um,
treatment? What's the treatment standard for the TZP in obese mice? Actually, we can't see that there will be some standards. It's based on the how much body weight loss you want to reach. Actually, we could choose different dose and different dosing frequency. But from our experience, because the TZP from 10 nanomolar, if we dosing them as a QD frequency, so around 2 weeks, it could reach a body weight loss around 20% to 30%. So it's based on how much body weight loss you want to reach. So if you found that maybe under this dosage or under this frequency, the mice the obese mice lost too much body weight, then maybe we could considering to decrease the dosage or decrease the dosing frequency. So actually, it's quite difficult to see whether there will be some standard for the TZP administration.
So kind of related, you were talking about body weight loss. There's a general question here about the muscle loss caused by GLP-1.
Yeah.
What are your kind of thoughts on this, and how does it come into play?
Actually, we have just give some examples, but maybe we could doing some combination used for some bimagrumab, or also we could combine with some different kinds of drug, like because we know there are some cleanest, there SNRA kinds of drug, their drug efficiency was on some uh muscle gain. So if we could doing some combination use, they will be help us to doing some relief or improvement on the muscle lean mass loss effect. So actually maybe for some combination use will be a good way to solve this problem.
Okay, we have a few more. There's one in the webinar chat about the administration of drugs.
See.
How do you administrate drugs?
What's the dosage of drug used based on what you choose? This also how do you measure? You check. Yeah, we do have the APOE the mass model. Uh, just how do you administer the drug? Actually, it's based on your drug priority. So if because we have some sub-Q, some oral, just it's based on your drug's priority. If your drug is some small molecule, and we could doing some oral, and if your drug is some antibody or some protein, we could do some sub-Q, and just it's based on different kinds of drug. What's the dose of drug used and based on what you choose the doses? So for the dosage choose, actually it's also based on the purpose of the drug efficiency study. So if you want to see some combinations, so maybe if you are giving different message could affect. Yeah, actually different administration route could affect the different bowel availabilities. Because for the oral or for the sub-Q one, they do shows different bowel availability. So maybe you could also doing some PK study and to see some bowel availability by different administration of the route. I think for the last question about the dose choose, yeah, actually it's based on the purpose of the study. Just because from several of our clients, our previous experience, some clients they want to do some combination use or they want to do a comparison between the positive drug and their own drug. So maybe I could give an example on some body weight loss study. Maybe just if you want to do some comparison, so we shouldn't doing some quite large body weight loss percentage. So that will means that they didn't show a good drug efficiency on some client's own drug. So actually just based on the purpose, yeah.
Okay, are there any more questions that people want to get in before we sign off today? Again, this webinar will be available on demand within a week on our website.
Yeah.
And you can definitely contact us at sales@gempharmatech.com if you have any more questions. Okay, it looks like we have one more. What is the main difference between HFD models and the GON diet model for MASH? Sorry, it's
Yeah, so for these two kinds of drug, they are all initial with some diet induction, but for the model established period, they are different. So for the HFD plus the CCL4, they will have a more shorter period. And for the gun diet, the Western diet, we need to just doing a quite long-term diet induction, so their model established time is different. And then it's also but for the Western diet, although it could have some more time of model established time, but actually this is more useful to mimic the clinical stages because for the clinical MASH patients, some of them they just get some unhealthy diet, so for the gun diet or the Western diet, they could have a mimic on this one. So it's based on the clinician's request. If they are a little bit hurried, then maybe we could using the high-fat diet plus the chemical toxin. But if the clinic is more focused on how much they have doing some mimic on the clean patients, then maybe we could using the gun diet induced one. And I think there was one question was the delivery of our
To Germany
mass to
and
Germany.
hygiene certificates.
Yeah. Actually, just maybe we could contact with our BD manager, or then maybe we could contact by email for this part because I'm not quite familiar with this delivery kinds of issues. So maybe
Yes,
our colleague will contact you later. Yeah.
we do have a German rep to
Mhm.
help you with those questions. The rep for Germany is what I mean. Um, well, thank you, everybody. Thanks for your good questions. Next month, we'll be presenting a webinar on respiratory disease models. So be on the lookout for promotion regarding that webinar on LinkedIn and through email. Again, if you have any questions, don't hesitate to reach out to sales@gempharmatech.com. Thanks for joining in today, and we'll see you next time.